Hello, bachcho. Let's see the question. So the question says a suitable vector for gene cloning in higher organisms is. So इन चारों ऑप्शंस में से हमें वो स्पीशीज आइडेंटिफाई करना है जो हायर ऑर्गेनिजम्स में जीन क्लोनिंग के लिए यूज किया जाता है. So the key concept in this question is to firstly understand the cloning vectors which are used for gene cloning or gene transfer in higher organisms like plants and higher animals. So the best cloning vector which is used for gene transfer in higher organisms is retroviruses. So retroviruses have RNA as their main genetic component. So retroviruses have this natural capacity or the natural capability to transfer their genetic component, that is the RNA, into their host cells. Host cells. So retroviruses transfer once attacks a host cell, then this retrovirus can transfer its RNA inside the host cell, where this RNA is converted into DNA. So this process of RNA conversion into DNA is done by an enzyme which is known as reverse transcriptase. So reverse transcriptase is an enzyme present in retroviruses which can reverse transcribe from RNA to DNA. So the conversion of RNA to DNA, and then this DNA is incorporated into the host DNA. So this results in the formation of the provirus. So now this proviral DNA, which has the DNA of the virus as well as of the host cell, with the DNA of the virus incorporated into the host cell, is the proviral DNA. Now this DNA can then be utilized for the formation or the of the viral proteins, and then these viral proteins are assembled together to form the virus, which is then helps in the replication of the virus. So the retroviruses can replicate in this matter by infecting their host cells. So in case of retroviruses, the gene of our interest can be encoded into the viral RNA. So once this viral RNA is infected into the host cell, then this viral RNA enters into the host cell where it is converted into DNA by reverse transcription, and this DNA is incorporated into the host cell. Therefore, in that way, the host cell now has the gene of our interest, because the viral DNA which was produced from the RNA was having the gene of our interest, and therefore the host cell now has the gene of interest which gets incorporated into it. So in this way, retroviruses are used as cloning vectors. Now, they are efficient cloning vectors for higher organisms because retroviruses infect higher organisms. So, the host for retroviruses are higher organisms like humans. And therefore, retroviruses can be used for cloning vectors or gene transfer in higher organism, because retrovirus infects these higher organisms and incorporates its DNA into the higher organism cell DNA. Therefore, retroviruses are good cloning vectors for gene transfer in higher organisms. So now if we come back to the question, then baculovirus is the first option. So this won't be the answer because baculovirus is not utilized for gene transfer in higher organisms, but instead it is only used in case of silkworms. Then we have retrovirus. So we studied that retrovirus are efficient cloning vectors for higher organisms. So this is our answer. The third option says Salmonella typhimurium. So Salmonella typhimurium is a pathogenic bacteria and it is not used for gene cloning in higher organisms. And the fourth one is Neurospora crassa. So Neurospora crassa is a red bread mold, which is a type of a fungus, and therefore it is not used as a gene cloning vector. So again, this is not the answer. So our final answer for this given question is option number 2, that is retrovirus. Hope you understood this well. Best of luck. Thank you.